Hi everyone. In this video, we'll consider the events that occur when an action potential arrives at a chemical synapse. So we'll redraw this neuron with all of our ion channels and everything one last time and sort of take notice of the fact that the action potential is propagating down the axon and simultaneously exchanging the various ions, sodium and potassium, at each sort of segment of the axon. And we'll notice that as the action potential arrives at the synaptic knob, the membrane in the vicinity is going to be depolarized, and this will cause the opening of calcium channels. And the calcium will rush in. The calcium is moving down its electrochemical gradient. So once the calcium rushes in, the calcium will interact with several proteins to cause a vesicle fusion with the presynaptic membrane. So let's redraw the synapse just a little bit bigger and take a closer look at the events that are occurring at this time. So we notice that on the postsynaptic membrane, we still have our sodium ligand-gated ion channels, and they also possess receptors for the specific neurotransmitter, the blue neurotransmitter that we're showing in this picture. We have our calcium channels, and on this presynaptic membrane, we have the what you could say is the docking station for the vesicles. So remembering all the principles of exocytosis will help to make sense of this process that's going to occur here. And we can see that we have a couple of different proteins that we want to pay attention to. So I'm only going to represent one neurotransmitter in the vesicle, but it's important to know that the vesicle contains multiple neurotransmitters. It's not just one. And I'm just going to make it a little bit easier to see. And then we'll take a look at, we have our V-snare proteins. Those are associated with the vesicle. We have our T-snare proteins. Those are associated with the docking station on the presynaptic membrane. And then we have one more thing called synaptotagmin, which is a protein that actually binds calcium. So as the calcium, we can see the calcium ions here, as the calcium rushes in, the calcium binds to the synaptotagmin, and that actually causes the vesicle to fuse. The V-snare and T-snare proteins twist up, and the vesicle sort of comes in, and then exocytoses the neurotransmitter into the synaptic cleft. So more than one vesicle will fuse at a time typically, and multiple neurotransmitters, as we said, are coming out of each vesicle. So a whole bunch of neurotransmitters will end up in the synaptic cleft. And we'll abbreviate neurotransmitter as NT. So neurotransmitter binds to the receptor on the postsynaptic membrane. And the binding of the neurotransmitter to the receptor causes a specific effect. In this particular case, it's the same as what we saw at the very beginning of our first neuron in the original drawing. The neurotransmitter binds, so sodium rushes in, and it's going to cause a graded potential in the postsynaptic neuron. So this time the potential will be excitatory, but remember that if it's a negative ion that flows in, it could be inhibitory. The one other thing I want to mention about this synapse is we highlighted previously that astrocytes actually sort of guard the gap where the synaptic cleft exists to prevent those neurotransmitters from escaping, and they also can serve as a location that can sort of manage the neurotransmitter after it's all finished at the receptor site. So there's a couple things that can happen to the neurotransmitter. The neurotransmitter can be broken down by enzymes, so sort of like chomped right off of that receptor site. It can just diffuse away from the receptor site. And then it can be, the word we use is reuptake. It could be taken back up into the presynaptic neuron and recycled, used again, or it could be actually taken up into the astrocyte. So there's a bunch of different options for the neurotransmitter, and it's going to vary by the specific neurotransmitter that we're referring to. So a couple things to keep in mind about what happens next. That graded potential that's going to occur in the next neuron, in that second neuron, will hopefully lead to an action potential, and that will follow all the way through to the next neuron and the next synapse. And as that neurotransmitter has potential to be recycled, the next time an action potential travels down this particular axon of that first neuron, we can use that neurotransmitter again, possibly if that's what happens to it, for the next set of events at the chemical synapse. So now at this point, we've covered everything that happens from generation of the action potential to completion of the action potential and communication with other neurons. So the one thing that I want to focus on a little bit before we move on from talking about the cells to talking about the bigger structures of the nervous system is the idea that different neurotransmitters exist and interact with different receptors. So we'll focus on that in our next video. That is all for now. Thanks for watching.